Good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD. So yes, so far we have dealt with living world, and from biological classification we have completed kingdom systems of classification, and we have started with the kingdom Monera, where we have completed the general characteristics, classification, nutrition and respiration. And now here we are to discuss about reproduction in bacteria. After this we will look into the kingdom Protista. This too we will exercise it today ma. In our next session we will finish off fungi and virus, viroids, prions, lichens, and then for whole chapter we will practice the EAPCET PYQs. Okay. So just focus on this. Reproduction in bacteria. Reproduction in bacteria is of two modes, asexual reproduction and sexual reproduction. Asexual reproduction, most common mode of asexual reproduction which takes place in favorable conditions is binary fission. Binary fission is the most common mode of asexual reproduction, and under unfavorable conditions endospores are formed. Okay, under unfavorable conditions endospores are formed, and sexual recombination is of three modes. One is conjugation, transformation and transduction. Conjugation, transformation and transduction. Here you see first is binary fission. Binary fission is the most common mode of asexual reproduction. Binary fission is most commonly called as amitosis because here both the karyokinesis and cytokinesis happens without the spindle fiber formation or without any requirement of spindle apparatus. Both karyokinesis and cytokinesis will happen, and one cell will divide into two without any spindle apparatus formation. That is why this is also called as what? Amitosis. That is all about binary fission. And asexual reproduction during unfavorable conditions is through what? Endospores. Endospores are released whenever there are unfavorable conditions. Endospores are released. Endospores have low water content and they have low metabolic activity. They are unharmed during pasteurization, that is heating at higher temperatures, and endospores are coated with many walls. See here you can see at the center there is DNA, right? At the center there is DNA which is covered by a core, and then above that it has a cortex. Above core, cortex is present. Cortex is made up of calcium dipicoleic acid. Remember, it is very important. Cortex layer is made up of calcium dipicoleic acid. Above cortex layer spore coat is present. Above all exosporium is present. Okay. DNA, core, outer membrane, above which cortex is present. Cortex is made up of calcium dipicoleic acid, spore coat and exosporium. This is the endospore that we have observed. Here it has what? Low water content, low metabolic activity, and this is unharmed during pasteurization. Endospores will act as means of perennation but not reproduction. Okay, endospores are what? Means of perennation but not reproduction. What does this sentence mean? Endospores are means of perennation means they will survive in the unfavorable conditions but they will not reproduce, they will not give rise to a new cell, okay, in the unfavorable conditions. In the unfavorable conditions they survive, that is why they are called as what? Means of perennation but not reproduction. Whenever the favorable conditions are resumed back, then one endospore will release out one bacteria. One endospore releases out one bacteria. Remember this, okay, out of one endospore coat which is covered by outer exosporium, spore coat and cortex, where cortex is made up of calcium dipicoleic acid. Here one endospore will release out one bacterium. Okay, one new bacterium will come from one endospore whenever the favorable conditions are resumed. These are the two modes of asexual reproduction that occur both in the favorable conditions and in the unfavorable conditions. Next here is the sexual recombination. Sexual recombination where there is no gamete formation, or gamete fusion occurs. That is why this is called as sexual recombination rather terming it as sexual reproduction. We use the term sexual recombination because there is no gamete formation or gamete fusion. Okay. How this happens? And sexual recombination occurs in three modes in the bacteria. First is conjugation, second is transduction and third will be transformation. First you see transformation. Transformation is identified by Frederick Griffith by his Griffith's experiment, and the model organism where he chosen is Streptococcus pneumonia. Streptococcus pneumonia has two strains, one is R strain which just has a wavy coat, and other is S strain which has a smooth coat over it. Okay, smooth coat over it, that is S strain. R strain has just wavy coat, S strain has a strong smooth coat over it, which gives shiny appearance. Okay, so this R strain bacteria, whenever it is entering the host immune system, this will be easily killed. R strain Streptococcus will be easily killed by the host immune system because it is not uncovered by a strong wall, strong cell wall. Whereas S strain cell wall is very strong, that is why it remains unharmed in the host immune system. It remains unharmed in the host immune system. Clear, right? R rough coat and S strain smooth coat. Clear, right, ma? Next. So here if you see transformation experiment, first S strain which is virulent, S strain which is virulent, which is injected to the mice, S strain virulent injected to the mice, mice will die because S strain has small coat, strong coat over it. R strain if you see wavy coat, when it is injected into the mice, mice will remain surviving, and R strain will be killed, okay, because it is not uncovered with a strong wall layer. Next here if you see in the third round where S strain is heat killed, where the wall layer is denaturated, S strain is heat killed and injected into the mice, mice will remain surviving. And in the fourth round what happens is living S strain is mixed with heat killed R strain, mice dies. So here in this fourth turn of this experiment, it was proved that there is something that is being, there is some particle that is being transferred from heat killed S strain to R strain and made R strain virulent. Okay, there existed what? Transforming principle. This is how transforming principle is discovered, wherein there is some particle that is being transferred from S strain to the living R strain bacteria, which made R strain virulent. Okay, so here there is no contact between donor and recipient. There is no contact between donor and recipient. And competence, what is competence? Ability to pick the DNA from the solution. Okay, here comes the term what? Competence. What is competence, ma? Ability to pick the DNA from a solution. Okay, so that is what? Transformation. That ability to transfer the DNA from the close proximity environment, taking up the DNA from the close proximity environment, or genetic transfer from environment to the bacteria is transformation, which is proved by what? Griffith's experiment. Second is what? Conjugation. Conjugation is discovered by Liderberg and Tatum, and the model organism is E. coli, which is the primitive type of DNA transfer. Conjugation is the primitive type of DNA transfer. So here we know bacteria has two genetic materials. One is called as genomic DNA and the other is called as non-genomic DNA or extra chromosomal DNA, which is nothing but the plasmid DNA. Plasmid DNA will be having what, beta? Factors. Factors like F factors, Ti factor, R factor, such factors will be present. If the plasmid DNA has F factor, this F factor is what? Fertility factor. Then this plasmid DNA, the plasmid DNA which has F factor, that bacteria is called as what? F plus strain. Okay, this is termed as what? F plus strain. Fine. F factor, that is termed as what? F plus strain. This through the sex pili or conjugation tube, it is being, the F factor is being transferred from the plasmid DNA to the plasmid DNA of the recipient. Those bacteria which doesn't have F factor is termed as F minus strain, and this is F plus strain. Okay, a conjugation tube or true pili or sex pili forms between F plus strain and F minus strain, and that is how F factor is being transferred from F plus strain plasmid DNA to F minus strain plasmid DNA. So at the end, the daughter strains, if you see, both F plus and F plus daughter strains are formed. If at all F factor is present in the genomic chromosome or genomic DNA, F factor is present in the genomic DNA, that is called as what? HFR strain. HFR strain. And the genetic material is transferred from HFR strain to F minus strain. Okay, genetic material is transferred from HFR strain to F minus strain. At the end, the daughter cells you can see is HFR and F plus. HFR and F plus strains are observed. Clear, right? Okay, this is all about conjugation. Next, be clear with the strains of F plus and F minus and HFR strain and F minus. Okay. Next, transduction. Transduction is discovered by the scientists Zinder and Lederberg, and the model organism here is Salmonella typhimurium. So here if the transfer of genetic material is from a bacteriophage to bacteria, then it is called as what? Transduction. Transduction, the transfer of genetic material is from bacteriophage to bacteria. Okay, bacteriophage to bacteria. Okay. Fine, this is what? Transduction, identified in Salmonella typhimurium. Now, useful and harmful bacteria. First we will go through all the useful bacteria. Making curd, which is widely known, that is lactobacillus, lactic acid bacteria, and then production of antibiotics by most of the gram negative bacteria like Bacillus subtilis or subtilin. Nitrogen fixation in the roots, Rhizobium and Frankia. Okay, both of them are symbiotic. Okay, so these are the useful bacteria. And harmful bacteria, if you see, cholera is caused by what? Vibrio cholerae, typhoid, Salmonella typhi. Tetanus is caused by Clostridium tetani. Citrus canker is caused by Xanthomonas citri. Okay, so these are the list of useful bacteria and this is harmful bacteria. And then Mycoplasma. Mycoplasma is the smallest living cell. It does not have any cell wall. These are known as what? Jokers of plant kingdom. They do not have any cell wall. They are pleomorphic. What is pleomorphic? They can change their shape or structure according to the environment in which they are inhabited. Like pleomorphic, that is why they are called as pleuropneumonia like organisms. Next, they are facultative anaerobes. Means most of the time they are aerobic. Whenever oxygen is not present, they act as anaerobes. Most of them are pathogenic to both animals and plants. Remember, these are the general characteristics of Mycoplasma, very important. All the terms, all the points are very important over here. Okay, smallest living cell, jokers of plant kingdom, its cell wall is absent, they are pleomorphic, they change their structure or shape according to the habitat they are inhabited to. They are called as pleuropneumonia like organisms, facultative anaerobes, and most of them are pathogenic to animals and plants. The examples being pathogenic to animals and plants that you have to remember. First is what? Which is broom, pleuropneumonia and Mycoplasma erythritis. These are the diseases which are caused by what? Mycoplasma. N number of times asked in your EAPCET exam. You can look into the PYQs. Okay, many at times asked question. Next is what ma? Actinomycetes. Actinomycetes is not there in your NCERT, it is there in your text, Telugu Academy. Important, specific for EAPCET. Okay, so here actinomycetes is what? This is branched filamentous bacteria radiating colonies in the culture. This radiate what? Colonies in the culture. Cell wall is what? Mycolic acid. Cell wall is mycolic acid. Mostly they are saprophytic and decomposers. Mostly they are saprophytic and decomposers. Actinomycetes, they are branched filamentous bacteria. They radiate colonies in the culture. Cell wall is made up of what? Cell wall is made up of mycolic acid. They are mostly saprophytic and decomposers. Clear, right? Actinomycetes. Next, parasites, Mycobacterium and Corynebacterium. Antibiotics, Streptomyces. These are the examples that you have to remember for sure. Okay, examples have to be remembered for sure. Okay, Actinomyces, general characters and examples. Clear ma? Now we will look into the kingdom Protista. Kingdom Protista includes what? All unicellular eukaryotes. Unicellular eukaryotes. So all unicellular eukaryotes are included. Kingdom Protista serves as common ancestor for 3 successive kingdoms. Those are, this is the common ancestor for 3 kingdoms, kingdom Fungi, Plantae and Animalia. These 2 sentences or statements are very important, can be asked in statement based questions. Okay, includes all the unicellular organisms and serves as common ancestor for these 3 kingdoms. One more statement based we can expect, that there are no proper boundaries of this kingdom. Boundaries of the kingdom Protista are not well defined. Boundaries of the kingdom Protista, they are not well defined. They form link with other kingdoms, that is Fungi, Plantae and Animalia. These 3 statements are unique and important for Protista, you have to remember. Next, so what are plant-like protists? Photosynthetic or plant-like protists, consumer decomposer fungi-like protists and animal-like protists or protozoans. These are 3 different types of protists that need to be highlighted. Now we will look into first general characteristics and then we will move on to photosynthetic, plant-like, fungi-like and animal-like protists. So here we go single-celled eukaryotes. Kingdom Protista includes what? Single-celled eukaryotes. They are primarily aquatic and they have membrane-bound organelles. Okay, membrane-bound organelles are present. They have well-defined nucleus and 80S cytoplasmic ribosomes are present. Whereas in the kingdom Monera, you can see 70S ribosomes, whereas here you can see 80S ribosomes with larger subunit of 60S and smaller subunit of 40S. Okay. Only 2 organelles which are in the eukaryotic cell, those are mitochondria and chloroplast. Mitochondria and chloroplast, both of them will possess what? 70S ribosomes. Rest all the cells, all the eukaryotic cell organelles might be, cells might be, they possess what? 80S ribosomes only. Only these 2 organelles possess 70S because they have come out of the prokaryotic cell. That is proved from what? Which theory? Endosymbiotic theory. Here you see motile structures. Motile structure in the kingdom Monera is only flagella. But here in eukaryotic cells, motile structures will be 2 like flagella and cilia. Both are the motile structures only. Flagella here is made up of the protein tubulin. Their prokaryotic flagella is made up of what protein? Beta flagellin. And if you have to see the microtubular arrangement in the kingdom Monera, flagella microtubular arrangement is 9+0, whereas the microtubular arrangement in the eukaryotic cell of Protista is 9+2. 9+2 microtubular arrangement, and these are the general characteristics. Okay, single celled eukaryotes, primary they are aquatic, membrane bound organelles, well defined nucleus, 80S cytoplasmic ribosomes, and flagella cilia will have what? 9+2 microtubular arrangement. Flagella is made up of tubulin protein. Next, here you see nutrition. Nutrition is holophytic, holozoic, mixotrophic, saprophytic and parasitic. These are different modes of nutrition. Reproduction you see both asexual reproduction and sexual reproduction. Okay, asexual reproduction is through what? Binary fission and spore formation. Sexual reproduction here in kingdom Protista, you can see cell fusion and zygote formation is observed. Okay, unlike sexual recombination which you see in the kingdom Monera, here you can observe what? Cell fusion and zygote formation. I hope the general characteristics are clear to everyone. Now you see the basic classification. First, photosynthetic or plant-like protists. Photosynthetic or plant-like protists includes what Chrysophytes, dinoflagellates and euglenoids, which includes only euglena. Next, consumer decomposer or fungi-like protists include what? Slime molds. We have 2 types of slime molds, cellular slime molds and acellular slime molds. And then animal-like protists or protozoans include amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. Clear, right? Next, photosynthetic protists. Photosynthetic protists, general characters you see, they have what? Protistian algae. Okay, Protistian algae, major portion, they are the major portion of phytoplanktons and they are the chief producers of oceans. These statements are very, very important, asked n number of times in statement based questions. Who are the chief producers in the oceans amongst all protists are photosynthetic protists or chrysophytes in specific diatoms. They are the chief producers of oceans and they form major portion of the phytoplankton. Now we will look into the general characteristics of chrysophytes. Chrysophytes include diatoms and desmites. Desmites are also called as golden algae. Okay, desmites are also called as golden algae. They are mostly aquatic, both freshwater and marine water. They are mostly aquatic and they are microscopic and they float passively in the water currents. Nutrition is, mode of nutrition is holophytic. Photosynthetic pigments that you observe in the chrysophytes or diatoms is what? Chlorophyll A, C, xanthophylls and carotenoids. Okay, chlorophyll A, C, xanthophylls and carotenoids. And cell wall, if you see, cell wall has silica, siliceous cell wall which is indestructible. And here if you see, there are two thin overlappings of cell wall above epitheca and below hypotheca. Epitheca and hypotheca together called as what? Frustule. This gives soap box like appearance. Okay, above epitheca and below hypotheca together called as what? Frustule. Clear, right ma? Clear with the general characteristics of chrysophytes, includes diatoms and desmites. Mostly they are freshwater and few of them are marine. Nutrition is holophytic and they are microscopic, they float passively in the water currents. Photosynthetic pigments include chlorophyll A, C, xanthophylls and carotenoids. Cell wall is siliceous, it has silica cell wall which is indestructible. It has two thin overlappings, epitheca and hypotheca together called as what? Frustule. Next, the siliceous cell wall which is over the diatom, when it is formed over the seabed, okay, that forms what? Diatomaceous earth which is called as Kesselger. Okay, all the diatoms which occupy over the seabed, the cell wall, siliceous cell wall will form what? Diatomaceous earth or Kesselger, which is again a major important point to be highlighted and remembered. They are used, they have a very good commercial importance. They are used in polishing, filtration of oils and syrups. Okay. Now, there is a classification of diatoms again based on symmetry. Based on symmetry, diatoms are again classified into centrales and pinnales. Centrales are those diatoms which are radially symmetrical. Centrales radially symmetrical, pinnales bilaterally symmetrical. Radially symmetrical, which means like the diatom is divided into any equal part by any plane. Okay, it is divided into two equal halves by any plane. Bilaterally symmetrical, it is divided into two equal halves by only single plane. Okay, centrales are radially symmetrical, pinnales bilaterally symmetrical. Remember that. Now, when it comes to reproduction, asexual mode of reproduction, most common, that is binary fission only for all unicellular organisms, be it prokaryotic or eukaryotic. Most common mode of asexual reproduction for unicellular organisms is binary fission. Sexual reproduction is through gamete fusion here in Protista. Gamete fusion occurs and they forms oxospores. Oxospores can be considered as zygote over here. But then oxospores are popularly known as rejuvenatory spores because oxospores will stay as such during unfavorable conditions and they will rejuvenate to the normal shape and structure. Normal structure whenever favorable conditions are resumed back. That is why they are called as what? Rejuvenatory spores. Important point and explanation, remember ma, we can expect in assertion reason. Why oxospores are called as rejuvenatory spores? Oxospores stay as such in the unfavorable conditions and they retain back the normal shape and structure whenever the favorable conditions are resumed back. This is all about chrysophytes. Now dinoflagellates. General characteristics of dinoflagellates include they are what? Golden brown algae, golden brown photosynthetic protists. Okay, dinoflagellates are golden brown photosynthetic protists. Unlike chrysophytes which are majorly freshwater inhabitants, these dinoflagellates are mostly marine. Okay, and if you have to see the photosynthetic pigments that are present in dinoflagellates, they include chlorophyll A, C, beta carotene and xanthophylls. Okay, photosynthetic pigments include chlorophyll A, C, beta carotene and xanthophylls. Next, cell wall of dinoflagellates have stiff cellulosic plates. Okay, they have stiff cellulosic plates on the outer surface. They have stiff cellulosic plates. And a nucleus here, if you can see, nucleus is mesokaryon. Why nucleus is termed as mesokaryon in Protista? Protista nucleus is what? Mesokaryon, because it has membrane bound nucleus, like how nucleus is present in a eukaryotic cell, membrane bound nucleus is present, membrane bound nucleus is present, whereas the division of the nucleus is without spindle fibers. So, it is exhibiting the nucleus nature of both eukaryotes and prokaryotes. That is why it is called as what? Mesokaryon. Very important point to be highlighted beta. Mesokaryon is the nuclear state. Nucleus is mesokaryon in Protista because it exhibits both the natures of prokaryotic nucleus and eukaryotic nucleus. Eukaryotic nucleus because it has a membrane bound nucleus, prokaryotic because it does not have any spindle fibers. Okay, spindle fibers are absent. Okay, that is amitosis. Next, flagella, you have two types of flagella. One is longitudinal flagella, second is what? Transverse flagella. Longitudinal flagella and transverse flagella. Two flagella are observed. So, at the center here there is a furrow. From this furrow, longitudinal flagella and a transverse flagella will arise. Both the flagella move as such, forming whirling whip movement. It gives what movement? Whirling whip movement. Two flagella, longitudinal and transverse, will move and it will give you whirling whips movement. And next, bioluminescence. Bioluminescence, these are what? Marine dinoflagellates, like for example Noctiluca, and red dinoflagellates which causes red tides in Mediterranean Sea. Same type like red tides in the Red Sea is also produced by a cyanobacteria, that is Trichodesmium erythraeum. Don't get confused between these two examples, that is Trichodesmium erythraeum and Goniolax. Okay, Goniolax is the Protista which forms red tides in the Red Sea. Okay, this is red dinoflagellate, and rapid multiplication or proliferation is done whenever there is rapid multiplication of or proliferation of this dinoflagellate in the sea. This will produce toxins. Those toxins are called as saxitoxins. This will kill the marine fish. When that fish is consumed by humans, it is even toxic to that. So, here this results in the formation of what? Red tides in the Mediterranean Sea. By what toxins is also important. Okay, you have to remember these two examples. Red tides in the Red Sea caused by cyanobacteria, that is Trichodesmium erythraeum, and a dinoflagellate, that is Goniolax. Okay, clear, right? Yeah. Here, third chrysophyte. Okay, third photosynthetic protist, that is Euglena or euglenoid. General characteristic, like if you see, they are mostly freshwater organism. Cell wall is absent in Euglena, important point. Okay, cell wall is absent in Euglena, whereas it is covered by a protein rich layer called as what? Pellicle. Cell wall is absent, but then it is covered by a protein rich layer called as pellicle. So here you can observe a paraflagellar body. The region where the flagella arises in the euglena is called as paraflagellar body, wherein a small flagella or a short flagella and a long flagella will arise. Okay, both the flagella will arise, and in euglena you can observe contractile vacuoles. Contractile vacuoles help in osmoregulation and excretion. Osmoregulation and excretion. And here you can see nucleus is completely membrane bound. Here you can observe short flagella and long flagella, which has small hair like structures like this. This is called as tychonematic flagella. Okay. So this is the general description of euglena, and the major characteristic feature of euglena is mixotrophic mode of nutrition. Mixotrophic mode of nutrition wherein in the presence of sunlight it acts as photosynthetic or autotroph. In the absence of sunlight it behaves as a heterotroph. So it is photosynthetic in the presence of sunlight, which is what mode of nutrition beta? Holophytic mode of nutrition. And photosynthetic pigments include what? Chlorophyll A, B, xanthophyll and beta carotene. Heterotrophic mode of nutrition or holozoic. Whenever when deprived of sunlight, whenever euglena is deprived of sunlight, these act as what? Saprophytes. This gets nourishment from dead and decaying organic matter. Okay, this gets nourishment from dead and decaying organic matter. So clear, right? Clear till here? Photosynthetic in the presence of sunlight, and these are the photosynthetic pigments. Heterotrophic or holozoic in the absence of sunlight, and what mode of heterotrophic nutrition ma? Saprophytic mode of nutrition. Next is reproduction. No sexual reproduction is seen, only asexual reproduction is observed. Asexual reproduction in the favorable condition is binary fission, and in euglena it is longitudinal binary fission. Longitudinal binary fission. Okay, and during unfavorable conditions, palmella stage is seen, cysts are formed for perennation. Perennation is for survival. And now fungi like protists. So here you have to remember, during favorable condition is euglena will reproduce by what? Longitudinal binary fission. During unfavorable conditions, that is by palmella stage, and cysts are formed for perennation. Fungi like protists. Fungi like protists include what? Slime molds. Slime molds are achlorophyllous saprophytic protists. Okay, slime molds are achlorophyllous saprophytic protists. The locomotion is through false feet called as pseudopodia. Locomotion or movement of amoeba, movement of slime molds is through false feet or pseudopodia. Okay, locomotion or movement of slime molds is through pseudopodia or false feet. Vegetative stage. Slime mold is usually slime mold has what? Multinucleated mass of protoplasm. This is surrounded by a plasma membrane. Cell wall is absent in slime molds. Cell wall is absent in slime molds. It has a multinucleated mass of protoplasm, which is syncytium condition. Syncytium condition. Okay, that is multinucleated condition. Okay, which is uncovered by cell membrane. Here we have two types of slime molds. One is cellular slime molds and the other is acellular slime molds. Cellular slime molds are formed by sequential karyokinesis and cytokinesis like this. Results in cellular slime molds. Whereas acellular slime molds, complete karyokinesis and later cytokinesis happens, which gives syncytium condition. You have to remember the examples of both cellular slime molds and acellular slime molds. So cellular slime molds examples include what ma? Dictyostelium and polyspondylium. Acellular slime molds, popular examples you already know, those include what? Physarum and Physarella. Okay, Physarum and Physarella. These are the examples of what? Acellular slime molds. So, acellular slime molds, cellular slime molds, and the general characteristics of slime mold is clear to you, right? Fungi like protists. Now, we will look into the reproduction. During favorable condition, what happens is beta, slime molds will extend their feet, that is false feet pseudopodia. They will extend their feet and they will form aggregations. Those aggregations are called as what? Plasmodium. They form aggregations called as plasmodium, which grow into several feet. Okay, that occurs during what? Favorable conditions. During unfavorable conditions, plasmodium will form what? Fruiting bodies. Plasmodium will form fruiting bodies. These fruiting bodies at the tips, they will release spores. These spores are transferred through what? Air currents. Okay, and spores will possess cellulosic cell walls. Slime mold doesn't have any cell wall, but the spores produced from slime molds possess cellulosic cell walls. Okay, during favorable conditions, plasmodium, they form fruiting bodies. At the tips, the spores are released. Spores have cell walls. Those are what? Cellulosic cell walls. Next is what? Protozoans. Protozoans are primitive relatives of animals. They are animal-like protists. Heterotrophs, they act as both predators and parasites. Cell wall is completely absent in protozoans. For example, here if you see amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. These are different types of protozoans that are observed. We will learn all the protozoans comparatively in a table. So, first here if you see amoeboid protozoans, entamoeba, histolytica or amoeba, anything you can take as example. Their wall is covered by what? Siliceous cell wall. They do possess the contractile vacuoles, food vacuole and a nucleus. Okay. Excuse. If you see ciliated protozoans, best example is paramecium. Paramecium do possess binucleate condition. Important. Binucleate condition where it has two types of nucleus, macronucleus and micronucleus. Okay, macronucleus and micronucleus. Macronucleus will look after the metabolic activities of the cell. Micronucleus will look after reproduction of paramecium. Important one, do remember macronucleus and micronucleus, and they have an oral groove through which food is ingested into the cell and cilia all over the body, cilia covered all over the body. They do possess contractile vacuoles as well. Next, flagellated protozoans, best example is trypanosoma, which causes sleeping sickness. Next is, last is what? Sporozoans, best example is what? Plasmodium. These are non-motile. Okay. We will learn all together comparatively. Look at here, amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. First if you see habitat. Okay, habitat. Habitat, amoeboid protozoans habitat includes fresh water, marine water and moist soil. Flagellated, just free living and aquatic. Ciliated protozoans, fresh water and marine. Sporozoans are endoparasites. Okay, sporozoans are endoparasites like plasmodium. Habit if you see, free living and all of them are parasitic only. Locomotion is through pseudopodia, that is false feet in amoeboid protozoans. In flagellated protozoans it is through flagella. In ciliated protozoans it is through cilia. Which is the fastest mode of locomotion? If that question is put, then your answer should be what? Cilia only. It is the fastest mode of locomotion. And sporozoans, the locomotion is absent. Distinct features if you see, marine forms silica shells, and ciliated protozoans, they have cavity or gullet for food transfer. Next, sporozoans are infectious spore-like stage. They give what? Infectious spore-like stage. Examples include for amoeboid protozoans, amoeba and entamoeba, flagellated protozoans trypanosoma, ciliated protozoans, paramecium, sporozoans, plasmodium. These are the examples. All examples are to be remembered for sure. Okay, this is all about the kingdom Protista, plant-like, fungi-like and animal-like. In our next class, we will be looking into fungi, classes of fungi in detail, Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. After that, most important segment of fungi is asexual spores and sexual spores, asexual reproduction and sexual reproduction, and life cycle of fungi. After that, we will be looking into a brief description of kingdom Plantae and Animalia. Anyhow, we will learn it in total in 2 separate chapters of plant kingdom and animal kingdom. After which, we will also look into those organisms which are not included in 5 kingdom system of classification, like virus, viroids, prions and lichens. After that, on total, we will practice the complete EAPCET PYQs of this chapter. Okay, so stay tuned and revise the class properly. If you are having any doubt, you can drop your doubts in the comment section. I will be addressing all your doubts. Thank you.